digestion, for your carbohydrate digestion, and also for your liver digestion. So what I recommend is that we're going to take a little bit of time to go over a couple of slides, or not a couple, a couple of slides. Introducing the endocrine system. We're going to start talking about a little bit of the endocrine activities and stuff like that. And then we're going to kind of, stop maybe a little bit, a third of a way through, go to lab. While you guys are doing your lab activities, and hopefully everybody can coordinate the lab activities so that when you incubate, your groups will incubate, all together, right? So then we'll have 90 minutes so I can finish up the lecture itself. Okay, that way we can get all this through. We don't have to be here until 8 for the lecture and then go in there and spend another hour and a half in the lab. We all want to go home, right? It's getting close to Thanksgiving. It's getting cold outside. Niners playing. We're gonna get away from that traffic by the time we get out. I don't know when they get out. They play at 5. Yeesh. Might as well stay until like 10:30. Just kidding. Okay, let's talk about the endocrine system. So with the endocrine system, this is just talking about glandular tissues that basically have the ability to make what we call hormones. And hormones are essentially just signaling molecules or compounds, mostly derived from proteins or amino acids or some kind of cholesterol compounds and things like that. But all of these particular particles that are made by these cells and secreted into the blood will act as a regulation to everything that's going on in the body in terms of metabolism, in terms of growth, in terms of repair, in terms of responding to other cells and tissues around you, and also for reproductive means as well. Okay. Now, of course, when we talk about the endocrine system, these glandular tissues, when they make their products, they secrete those products directly into the blood, right? That's a big difference between endocrine versus exocrine, right? We're talking about exocrine like your pancreas secreting digestive enzymes into the small intestine, your salivary gland secreting saliva and digestive enzymes into the oral cavity, your lacrimal gland secreting tears out into your eyes and stuff like that. Those are all exocrine glands, like sweat glands secreting sweat onto the surface of your body, right? All of those are exocrine glands. For endocrine glands, we're talking about things like, you guys remember we talking about gastrin, right? Being secreted by the G cells, secreted directly into the blood. Therefore, it becomes an endocrine type of tissue, stuff like that, okay? So everything that we talk about today is going to be referring to the endocrine activities of your body. And the endocrine system is composed mainly of these, what we call major endocrine glands that are part of this important extrinsic regulation of homeostasis. So the other extrinsic regulation of homeostasis that we have will be our nervous system, which is a more rapid way to basically communicate throughout the whole body. With the endocrine system, it takes a little bit longer because hormone has to be secreted, and therefore when the hormones actually do stimulate those target tissues, they actually have a longer lasting effect compared to the nervous system itself. Okay? So here's some major endocrine glands that we're going to be referring to. Some of the major ones basically are located within the central nervous system, so in the brain area. So you have like the pineal gland, you have the hypothalamus, you have the pituitary gland, and then down in front of your neck you have the thyroid gland. Behind your thyroid glands you're going to have 4 nodules of parathyroid glands, and then below that, supposedly you have a thymus gland here, but what I'm going to talk about is located in the anterior and mediastinum, right in front of your heart. But as an adult, we no longer have that glandular tissue, so we're not going to really talk about that. Down here in the abdominal cavity, located on top of your kidneys, we have what we call the adrenal glands. And then in the abdominal cavity itself, we would have that pancreas. You guys remember the pancreas, a very important exocrine structure, right? That secretes the vast majority of your digestive enzymes. But there are a collection of cells within the pancreas that is responsible for secreting hormones that will tightly regulate the amount of glucose inside your blood. Okay? And as far as reproduction goes, the reproductive process of both males and females individuals are regulated tightly by the endocrine system as well. Okay? So the other 2 endocrine tissue in the males are the testes, and in the females are the ovaries as well. Okay? So we'll talk about all of these guys. Here's just a short list of all the glandular tissue that have potential endocrinic activities, right? If you can think about other type of tissue that we already talked about, like in the kidneys, which ones have endocrinic activities, in the small intestine, which cells has endocrinic activities, in the heart itself, there's certain cells that can also secrete hormones that can regulate some of these particular processes as well, okay? Make sure you make that list. If you guys don't remember the list that I mentioned about, I'll write it down for you guys one more time. Okay, so for every hormone that we talk about, obviously identify that hormone. Okay, and then for every single hormone, there has to be a source that produced that hormone, right? So indicate the source. And then for every single source, you have to have some kind of stimulatory factor. So what stimulates the source to secrete the hormone? So stimulatory factors. Well, I guess you can put inhibitory factors as well. So what are these stimulatory and inhibitory factors that act on this source? Wherever that source is, it's a glandular tissue, right? And what does this glandular tissue do in response to these? Let's say if it's a stimulatory source, well, once these particular sources are stimulated, what kind of hormones do they secrete? And if they secrete those hormones, where do they go to? So what's the target of those hormones, or targets? Because some of these hormones have multiple targets that they bind to. Once they get to those targets, what are their individual functions at each of those targets? So it depends on the targets itself, the functions may slightly differ. Okay, it depends on where it is that you have these targets. So make sure you have that list, and make that list for every single hormone that we talked about. So you guys should already have made that list for renin, the angiotensin 2, for aldosterone, for antidiuretic hormone. If not, it's okay, I'll reiterate some of that particular hormones. You should already have the atrial natriuretic peptide. That's one of those particular hormones secreted there. You also should have like cholecystokinin. You should also have secretin already listed down in that list, right? If you haven't made that list yet, it's all right. Can't make you make a list, though. Just as long as you know it. So you can do well on the exam. Because the exam's gonna focus a lot on this endocrine stuff too, okay? Since the endocrine system is the endocrine system, and the reproductive system is heavily relying upon the endocrine activities, that's what the hormones and all that regulatory activities. All right, so different classification of hormones. You guys don't have to worry about these classifications, right? If you want to read it, it's there for you, because different hormones are made by different sources and stuff like that. But what you do need to know is this. So hormones come in two basic varieties. One is that overall structure of that hormone makes it polar, which means they love to interact with the water environment and stuff like that. They're partially charged, so therefore they don't mind interacting with the charged water environment. Otherwise, other hormones are what we call non-polar. And non-polar are hormones that basically, when they come together, they are non-polar. They don't have any charges associated whatsoever, right? So therefore, just kind of break down these two categories, right? As we talk about some of these, I'm not gonna list every single hormone and categorize them for you, but essentially, if the hormones are not steroid hormones, or they are not melatonin, then they are not non-polar, okay? Or sorry, yeah, if they're not steroid, so if they're steroid hormones, let's put it another way. If they're steroid hormones, or if they're melatonin, that's one of the hormones, then they are non-polar. If they don't fit these two categories, they are now polar. Okay, so I just moved thyroid hormone up here to the polar category. So basically, if you have that on your note, just make that switch, right? Just circle it, bring it up to the polar category. Now, keep in mind that every single hormones, when they are secreted out into the circulation, and as they reach that target tissue, they have to bind to that target tissue in order to stimulate the reaction or function, intended functions that they have. The binding of the hormones is to their specific receptors. And we understand receptors are just, proteins that are made by those particular target cells, right? Now, it all depends on the type of hormones, because for polar hormones, since they are partially charged, they cannot move across that membrane. So all polar hormones must bind to their specific receptors located on the surface of those target cells, on the cell membrane. So therefore, those receptors are integral proteins inserted onto the membrane of those target cells that will specifically bind to those hormones that are considered to be polar. Okay? For non-polar hormones, because they are non-polar, they can easily move across that membrane via diffusion. Right? We talk about simple diffusion based on the concentration gradient. In a biological system, that cell membrane is always permeable to non-polar entity. Okay? So therefore, any non-polar hormone can easily move across the cell membrane of their target cells. But once they get into the cytoplasm, they will have to bind to the receptors specific for those hormones located either in the cytoplasm and or inside the nucleus. Okay? So therefore, all hormones must bind to some kind of receptors. It depends on the class of hormones. The receptor's location can be different. So for polar hormones, their receptors are located on the cell membrane. For non-polar hormones, their receptors are located within those target cells, inside the cytoplasm and or the nucleus. Okay. And that's just something just to kind of keep in the back of your mind. These terms pro-hormones or pre-hormones, because every single hormones that are made in the body is made by a sequence of amino acids all strung together, or whatever it is, however they're expressed. But when they're made as a big old strand, there could be multiple things attached to them. There could be regulatory proteins also attached to them. So therefore, when they're first made, they're usually made in an inactive form, right? And then further down the line, they will get cut into the active portion of that hormone. So therefore, pre-hormones or pro-hormones is just a way for our body to tightly regulate the activities of these hormones before they reach their target cells. Okay? Because you don't want them to be activated immediately as soon as they're made. What if they don't ever reach their target cell? What if they bind to something else that looks similar to those receptors? Then they may inadvertently activate another cell, right, to do what those cells are not supposed to do. So therefore, this is just a way to help regulate the fact that some of these hormones, when they're first made, they're made as a string of other things that are compounded together. So once they reach their target cell or in transit, they will be cut into their active portion of that hormone. Okay? So that's what pro-hormones or pre-hormones refers to. All right. Now, like I said before, every single hormone has to interact with their specific receptors. Okay? Again, once they bind to their specific receptor, they will set off a series of cellular activities that will allow for those cells to react appropriately to whatever these hormones are. Now, the way a target cell responds to the hormone can be multiple ways. Okay? And these are listed here for you. So the hormones can act on the cell in what we call an antagonistic way. The hormones can act on the cells in what we call a synergistic way, or the hormones can act on the cell in a way we call a permissive type of activities. Right? So synergistic basically refers to the fact that you have two different hormones that will bind to the same cell, and basically through the effect of each individual hormone, they enhance the overall effect of that cell. Okay, so whatever this hormone is and the other hormone, they bind to different receptors on that cell, but as they bind to the cells individually, they allow for that cell to respond in a way where the effect is additive. Okay? So therefore, we call this a synergistic effect. So for example, sort of like milk requiring estrogen and stuff like that, prolactin and oxytocin, so all three of these hormones act on the mammary gland to enhance the effect of milk production and all that good stuff. All right. The second type of activities that's imposed by the hormones at these particular target cells is what we call permissive effect. Permissive effect means that one hormone is going to stimulate that target cell to become sensitive to another hormone that will be secreted out later on. Okay? Because now both of these hormones will act on the same target. However, this hormone may act on the same target before the other one, so that this target can become more sensitive to the other hormone, basically priming it or permitting it to become more reactive to the second hormone, right? So for example, in the female reproductive system, at the uterine lining, like inside the uterus, there's a layer where it grows and stuff like that to accommodate for pregnancy and things like that. So during certain cycle or times of the cycle, there's a first hormone we call estrogen, which stimulates that lining of the uterus to grow massively, just in preparation for implantation of the fertilized eggs if you do fertilize that particular oocyte, right? So therefore, as estrogen binds to that particular layer of tissue, it allows for that tissue to now express receptors for another hormone we call progesterone. So it's kind of priming or making that layer of tissue more and more sensitive to progesterone, which comes later in the secretion. So therefore, as the female individual release now progesterone later on, but the progesterone can bind to those receptors on the same tissue and enhance the activities of that tissue even more, right? Because progesterone is one of the more important type of hormones that will help to regulate the early stages of pregnancy as well. Okay? So that's just what we call a permissive effect. Now, on the other hand, if you do have two different hormones acting on, let's say, a very similar area of the body, they may usually have what we call antagonistic effect. Antagonistic effect means that these two hormones will have the opposite effect on the same target. Okay? So for example, in the pancreas, you have these two hormones called glucagon and insulin being secreted by the cells of the pancreas. Both of these hormones will help tightly regulate the blood sugar. However, let's say when it acts on the liver, insulin stimulates the liver to pick up the glucose from the blood and stores that glucose. Whereas when glucagon acts on the liver, it stimulates the liver to break down the glucose from the glycogen and release it back out into the circulation. Okay? So they have opposite effects of each other. All right, now under normal physiological conditions, when an endocrine gland secretes their hormones, they will always secrete it in a certain amount. Whatever that original amount is, we can chart that and we can measure how long the body takes to break down the content of those active hormones into half of its original concentration. So let's say if I secrete out 100 milligrams of a hormone, and it takes 30 minutes for the body to adequately break down all of these active hormones to about 50 milligrams. Okay? So the time that it takes for the body to reduce the original concentration of whatever it is down by 50% is referred to as the half-life, okay, of any substance. Once you guys get into nursing programs and stuff like that, you will have to measure the half-life of any kind of medications that you're about to give to your patients, because that half-life allows you to understand when the next dose of medication must be given, so that you can maintain what we call a therapeutic dose in order for that medication to help the patient recover from whatever condition that ails the patient, right? So same thing, let's say if you guys are taking a medication, right? Let's say we got some kind of bacterial infection, right? And then we go to the physician's office, they give us a prescription of antibiotics, and you go fill that antibiotics at the pharmacy, and the pharmacist says, all right, well, take this pill 3 times a day, okay? So what's a day? 24 hours, right? Divide by 3, that's 8 hours span. Makes sense? So therefore, you have to take that pill every 8 hours, right? Why is that? Well, because within 8 hours, the original concentration of that medication will have been reduced by half, which means it's no longer therapeutic. It's no longer working to protect you to fight off whatever this particular infection is. So therefore, at the 8-hour mark, you have to take another pill so you can raise that therapeutic dose back up to what it should be, so that it can protect you, right? And then the next 8, you can go again through the next 8 hours, and then at that time, you take another pill. So basically, that whenever you have to take a pill, that marks the half-life of that particular concentration of medication. Okay, so if they tell you to take a pill once a day, what's the half-life of that pill? 24 hours, right? Because a day has a 24-hour cycle, right? Unless you live in some other distant planet that we don't know about.
Do they wait until it drops to half concentration?
They usually do. Because if you give too much of it, what happens is that you have toxicity of that medication. Because if you don't adequately break down the active ingredients of those medications, then you have too much of it, it can cause side effects, right? Harm to other, tissues, and especially harm to your liver, harm to your kidneys and stuff like that, right? So therefore, there's certain medication. Well, every single medication that you give to your patients will be broken down by 2 organs of the body. the liver, and the kidneys. That's it. So therefore, for every single medications that you give your patients, if you know it will be broken down by your liver, before you administer that to your patient, you need to check the patient's liver function. Because if the patient's liver is not as highly functional or normally functional, and you give this high dose of that medication, it actually will overwhelm the body and cause a lot of different side effects, right? And it will cause the liver to basically become even more injured, because now you overwhelm the liver and stuff like that. And of course, if you have a medication that has to be broken down by the kidneys, then you need to do what we call a renal function test or a kidney function test before you administer that medication as well. Okay, so please always keep that in mind, because whatever we do, we protect the patient first and foremost. Okay. So hormone half-life is just basically indicating the time that it takes for whatever that concentration or active concentration of hormone that is secreted out in the first place to be reduced by half its original concentration. So if I'm an endocrine gland and every single time I'm stimulated, I will always secrete out 100 milligrams of whatever that hormone is, and it takes about 10 minutes for the body to break down those hormones to reduce that 100 milligram to about 50 milligram, then the half-life of those hormones is about 10 minutes. Make sense? So there are hormones of the body that has very long half-life, and there's hormones of the body that has a very short half-life. There's some hormones that basically persist for a couple minutes and it's done. And so therefore those glands have to continue to secrete that every single time it reaches that half-life. Unless you've already finished doing what you need to do that is regulated by those hormones. Okay, because remember, the hormones is not medication and stuff like that. The hormones are secreted out as a mean of communication, as a mean of regulation of metabolism and regulation of homeostasis. Okay. So, all right. Oh, another thing to think about when you guys are full-fledged clinicians is that for certain medication, there could be interactions with other medications as well. Right? We're not going to get into that discussion. That's more like toxicology and stuff like that. But a lot of this drug interaction is not due to another drug that the patient is also consuming. This can also be due to herbal medication that the patient is consuming, which the patient will not tell you, because the patient will not think that drinking tea or taking ashwagandha or taking some kind of licorice root or drinking ginseng and stuff like that is medication. So they will not tell you that they're doing that. Okay, so you have to ask them. Right? Or you have to tell them, all right, when you take this medication, please don't eat grapefruits or drink any grapefruit products with this medication. Because if you do this, it will interact with this medication and causes your liver to basically be injured, okay, or things like that. Or it limits the metabolism of your liver by breaking down these products, and therefore you will accumulate too much of this medication and induce what we call toxicity, which causes damages to other cells and tissues of the body. So that's something we all have to worry about as healthcare professionals, right? That's one of the hardest things to worry about and stuff like that. Okay, now some of these hormones have what we call priming effects or upregulation effect, which basically means that it will stimulate. It's just like that permissive type of activities. It will stimulate whatever that target cell is to increase its sensitivity to another hormone or maybe to the same hormone and stuff like that, right? So whatever the case is, if you stimulate a target cell by employing a hormone to stimulate that target cell, the hormone can stimulate that target cell to increase the amount of receptors for whatever else, therefore causing what we call a priming effect in those target cells to whatever else that is supposed to be coming to bind to those particular receptors later on. So basically, some of these hormones have what we call a sensitization effect, right, to those target cells, allowing for those target cells to increase the number of receptors to whatever it could be, like another hormone or some other ligand or whatever it is, to allow that cell to become more sensitive to another signal, right, to another stimulus. Okay, so that's what we call a priming effect or an upregulation effect. On the other hand, hormones may also have what we call a desensitization or a downregulation effect. So in these particular cases, if you release too much of those hormones for any particular reason, maybe because you have some kind of tumor in those particular glandular tissue that constantly secrete too much of those hormones, or if you're taking exogenous hormonal medications, like some kind of pills that contain those hormones and stuff like that, if you have too much of those, what happens is that those particular cells and tissues become desensitized to the effect of that hormone. Okay, it all depends on what kind of activities you want to impose onto that hormone. So, if you have too much of those hormones lingering around for any particular reason, maybe because whatever that glandular tissue is that is responsible for secreting those hormones are always being stimulated by something. So therefore, there's too much of those hormones. Well, then the cells, the target cells that receive those hormones will say, you know what? We don't need to be as sensitive to this hormone. There's so much of them lying around or always available to us. So, and we don't want to constantly go through these activities that's stimulated by these hormones. So what we're gonna do is we're gonna take away some of these receptors from the surface of our cells to prevent that hormone from causing us to react too aggressively all the time. Does that make sense? Because it's no longer homeostatic, okay? So in those particular cases, you may have cells that become what we call desensitized to the effects of those hormones. Like case in point, if you have too much particular blood glucose or whatever the case is, but for some reason your cells can't pick up those glucose, so therefore maybe you don't have enough or you have too much insulin or whatever the case is, the cell become desensitized to the effect of that particular hormone. Okay? And they no longer will react as appropriately, as normally as they should to that hormone. Okay? That may result in things like type 2 diabetes. So therefore, oftentimes in the human body under normal physiological conditions, unless you have some kind of underlying medical conditions, right? So let's say talking about normal activities overall, first and foremost, every single glandular tissue that secretes hormone will always secrete it out in a definitive physiological concentration. So it knows how much to secrete out every single time, okay? Now, of course, it does that, but then if you overstimulate it or you understimulate it, then it will secrete more or less than what it needs to. And if that persists for a long period of time, then it becomes pathological. It's no longer physiological, okay? So therefore, whatever the case is, we have to address the problem and fix that so that everything can be maintained as a balanced, activities. So for every single endocrine gland under normal situation, they will always secrete out a finite amount of concentration of those hormones all the time, just to achieve what they need to achieve. Secondly, to prevent this down-regulation or to prevent the tissue from becoming desensitized to the effects of those hormones, oftentimes the endocrine gland that secretes the hormone will secrete this hormone out in what we call a pulsatile fashion. So it secretes it out and then it stops. And then when the level starts to go down again, then it gets stimulated to secrete it out again. So that's why the hormone half-life is also very important. Okay. All right. So let's talk a little bit about how these particular hormones work. Like I said before, right? Every single hormone must bind to their receptors in order for them to stimulate those target cells to do whatever they need to do. So just keep in mind the two classifications of hormones, either they're polar hormones or non-polar hormones, okay? So for non-polar hormones, they will bind to their receptors located within the cytoplasm and or nucleus of those target cells. And those receptors come in a complex structure, and together we call those the nuclear hormone receptors. So there's a class of receptors that binds to non-polar type of hormones we call the nuclear hormone receptors, because essentially those complex structures will move into the nucleus eventually once they bind to those hormones. Okay. For polar hormones, they will bind to their receptors located on the cell membrane. Right? And for every single hormone, there is a specific hormone receptors for them. And they will bind with those receptors with relatively high affinity and high specificity. So let's first talk about the nuclear hormone receptors, these particular receptors that are specific for non-polar hormones. Okay? Now, for every single nuclear hormone receptors, there's two regions that make up this particular structure. There's a region we call the ligand binding domain, which is a region of that receptor that binds to the hormone, right? That has a high affinity for that hormone and allow for the exact fit of those hormones to that particular receptor. And that ligand binding domain is actually going to be attached to another portion called the DNA binding domain. Okay? So there's two things attached together. One allows this receptor to bind to the hormone. The other one is going to become activated after the binding of that ligand in order for it to bind to the DNA. Hence the DNA binding domain. Okay? Now, once the ligand binding domain is bound to that specific hormone, like I said, it activates the DNA binding domain, and the whole complex will be transported into the nucleus of that target cell. Once inside the nucleus of that target cell, it will bind to a DNA strand or a segment of DNA around where the gene is that they want to stimulate, and that portion of the DNA is referred to as the hormone response element. And then something will happen along the way, which I'll show you guys the picture in a second. All right, so here's a picture. You guys can go back and read this after. So here's a picture that shows you how it works. So imagine if you have a steroid hormone, a non-polar hormone of some sort, right? That non-polar hormone is being transported by a carrier protein within the blood. Okay, because first of all, they're the non-polar hormone. So they can't just float around freely inside the circulation since plasma is water and water is polar. So therefore, you have these non-polar hormones bound to all of these carrier proteins which transport them safely within the circulation itself. Once they reach their target cell, then they release the hormones, and the hormone can easily translocate into the cytoplasm, which means they can easily move across that membrane, right? There's nothing that needs to stop them from moving. There's nothing that needs to help them from moving across this membrane. All right, so once they move into the cytoplasm of this target cell, they will bind to the ligand binding domain of this overall nuclear hormone receptor. So this whole complex here is referred to as the nuclear hormone receptor. Okay? So for this portion here called the ligand binding domain, it is specific for this type of non-polar hormone. So for every single non-polar hormone, there is a specific type of ligand binding domain for those particular hormones. Okay? All right, once that hormone binds to the ligand binding domain, it activates the DNA binding domain, which is this portion here, and the whole complex moves into the nucleus of that cell. Once it gets into the nucleus of that cell, it's going to look for the DNA strand. And imagine here's a DNA strand. And on the DNA strand, here's the target gene that it wants to activate. But in front of that gene, there's a sequence of DNA we call the hormone response element. And the hormone response element actually has two different sites. And these are called half sites because each half site will accommodate a complex of that DNA binding domain, ligand binding domain, and that hormone that's bound to that ligand binding domain. Okay? So now you have this whole complex here with the hormone bound to this ligand binding domain, translocating or moving into or transported into the nucleus. The whole thing is going to bind to one half site of that hormone response element. The other half site will accommodate another complex that has the same hormone bound to the ligand binding domain. Does that make sense? Okay. So now you have two of these complexes containing two of these similar hormones, both occupying each half site. Once you have two of these complexes occupying the half site of that hormone response element, this complex will become a dimer. It comes together. It dimerizes. Now, this dimer is referred to as a homodimer because of the fact that the two hormones that is bound to the ligand binding domain are the same type of hormone. Okay? We're not talking about like, oh, they're both nonpolar. No, they are both of the same hormone. They're obviously both nonpolar, but they're both similar hormones, right? So let's say for aldosterone, aldosterone is a nonpolar hormone. So therefore, if this steroid hormone was aldosterone, then this other steroid hormone is also aldosterone. Okay, it's the same hormone. It's not a different hormone of the same class, right? So therefore, because of that, we call this dimer a homodimer. And once this dimer is formed, it actually becomes what we call a transcription factor. So it increases the activities of this nucleus, stimulate those enzymes so that they can easily and quickly copy this DNA strand, this gene. And why do they need to copy this gene? So they can make a messenger RNA, so that they can basically take that messenger RNA and translate it into some kind of protein structure. Okay, whatever that protein structure is that was needed to be made and is stimulated by these hormones so that the cell can make those particular proteins. Okay, so let's say for aldosterone, for example, right? Let's say if we're using that particular activities or using that particular example of the hormone, when aldosterone forms a dimer and stimulate the transcription of this gene, what do you think that gene codes for? What's the function of aldosterone?
Aldosterone increases reabsorption.
Okay, reabsorption of what?
Of water.
Small ions.
Not water.
Sodium chloride.
Sodium ions, chloride ions, right? So therefore, on the cells, on the apical surface, the top surface of the cells of the distal convoluted tubules, you don't have those ion channels. Not yet. But once aldosterone is secreted and stimulate the distal convoluted tubular cells, they will quickly make those ion channels and they will express those onto the surface of their cells, so that they can bring in the sodium ions and chloride ions and whatever else, right? Otherwise, how would it do that? Does that make sense? Yeah? No? Yeah. Good. So just think of it that way. Now, for thyroid hormone is a polar hormone. However, it does have very similar activities to the steroid hormone in that their receptors, they're polar, but their receptors, for some reason, is actually located all the way in the nucleus of the cell. The receptors for the thyroid hormone is not located on the cell membrane itself. So thyroid hormone has to basically get into the cell in order for it to bind to their receptors. Okay? So imagine here's thyroid hormone. And thyroid hormone, there's two different variations of thyroid hormone, which we'll talk about later on. There's a variation of that we call T4, which is tetraiodothyroxine, which is basically a thyroid hormone that is bound to 4 iodine compounds. Okay? So that T4 is the inactive form of thyroid hormone. The active form of thyroid hormone is called T3, which means it's a thyroid hormone that is bound to only 3 iodine. Okay? So therefore, T4, let's say being transported in the blood, in its inactive form, bound to this particular carrier protein, being transported all the way to this target cell. T4 then is released, and in order for T4 to be transported, it has to bind to some kind of carrier protein here, which is not shown in this picture. So there's a transport protein that binds to T4 and allows it to enter this particular cell through what we call facilitated diffusion, right? Of course, because T4 is a polar compound. So once T4 gets into this particular cytoplasm, there's another enzyme that picks up T4 and removes 1 iodine from T4 to convert that into T3. And then that same enzyme is going to transport T3 all the way to the nucleus and release it so that it can enter the nucleus of that particular target cell. Okay. Once inside the nucleus, there's a receptor portion, there's a ligand binding portion on this nuclear hormone receptor specific for T3. Okay, so we call this the thyroid receptor, basically. Okay. Now, this thyroid receptor, once it binds to T3, it's going to bind to this half site on this DNA. The other half site is going to accommodate another complex of nuclear hormone receptor. However, the other complex of nuclear hormone receptor, its ligand binding domain, is not for T3, but it's for another compound we call 9-cis-retinoic acid, which essentially comes from vitamin A. Okay? So therefore, you have this particular nuclear hormone receptor that is specific for 9-cis-retinoic acid occupying the half site on this hormone response element. And you have a nuclear hormone receptor specific for T3. Okay, they both bind to each of these half sites. Now the half sites here, both of these represents the DNA strand that accommodate this structure, the nuclear hormone receptors, for thyroid hormones. So instead of calling it a hormone response element, we call this a thyroid response element. Just basically the same thing, just substitute the term hormone for a more specific hormone we call thyroid hormone. Okay? So therefore, thyroid response element represents these two half sites. One of the half sites will accommodate for this nuclear hormone receptor that is bound to T3. The other half site accommodate for a nuclear hormone receptor that is bound to 9-cis-retinoic acid. Okay. Both of these, once they are completely bound to each half site, they will become a dimer that comes together. Now, because this dimer is composed of two different types of ligands, one is C3, the other one is 9-cis-retinoic acid, we call this a heterodimer. Now, this dimer, once it's formed, also acts as a transcription factor. And this transcription factor is going to activate the enzymes necessary to transcribe this gene so that you can make a bunch of messenger RNAs, so you can translate those messenger RNAs to make whatever necessary proteins that this thyroid hormone was designed to stimulate in this particular cell. Okay? And keep in mind, when we're talking about thyroid hormone, it is the primary metabolic hormone of the body. Okay? If you don't have adequate amount of thyroid hormone being secreted out, then you will feel absolutely fatigued. And it's not like you lack motivation, you just can't do anything. Okay? Even if you want to, you can't do it. You just simply don't have the energy to do anything. So oftentimes, with patients with hypothyroidism, patients that have very low thyroid hormones, when you look at them clinically, they look like they have major depression because they have no motivation to do anything. They just want to sit around and they can barely talk. If you talk to them, they can barely get words out, right? And you can barely understand anything that they say because it's just, they don't have any energy to even move the muscles to allow them to basically vocalize whatever words they're trying to express. They have full cognition. They can think, they can understand everything that's going on. They just can't physically do anything at all. Okay? So therefore, a lot of those patients get mistreated and is given antidepressant medication, which worsen their issues. Okay? If you don't have adequate amount of thyroid hormone, it's very bad. You may die. Make sure you check the thyroid hormones in any patients that are brought into your clinic with major depressive symptoms. Okay? Because you don't want to misdiagnose and mistreat your patients in that way. It's easy. Just take a blood test, look for thyroid hormones. If it's normal, okay, fine. Maybe treat depression, because that could be the reason why the patient has depression. Or at least the patient clinically looks like they are depressed. All right. Any questions about the non-polar hormone activities and thyroid hormone activities? Okay. Make sure you guys know those. You guys just had a quiz today, right? Okay, good. For quiz number 8, it'll be in class. Okay, so don't be late. It'll be like the only time I give you a paper quiz, just to see if you guys still remember what that feels like. Actual physical piece of paper. Let's see, test your writing skill, your grammar skills, your English skills all together, because ever since the online format and the COVID and stuff like that, I've been finding it very, very difficult to grade essays, because I would just tear up that essay before the second sentence. It's so bad that people just don't know how to write anymore. It's crazy. And we're talking about my graduate students, right? That's depressing. So we'll see how it goes with you guys. Maybe it's just one question. That's worth 20 points. It'll be fun. All right, anyhow. So let's talk about the polar hormones. So for the polar hormones, because they cannot cross that cell membrane, they have to bind to what we call the receptor proteins specific for those particular hormones. So therefore, every single hormone that is a polar hormone typically utilizes what we call secondary messengers. Okay? And secondary messengers is a great way for those target cells to tightly regulate the activities of those hormones. Because you don't want those hormones as soon as they bind to stimulate something right away. But you want to have these other regulatory pathways to basically say, all right, well, I have these hormones binding to me, and it's going to initiate some kind of event inside the cell, but I can also regulate some of those events as well, so that the overall outcome of it is what I want it to be, instead of just immediately getting, a full-blown effect from those particular hormones themselves. Okay, so therefore, all of these particular type of receptors that bind to polar hormones is going to utilize some kind of secondary messenger inside their cells, so that they can also help to regulate some of the processes that's stimulated by those hormones. So the three type of secondary messengers is known as cyclic AMP, using this enzyme called adenylase cyclase, calcium calmodulin, using the enzyme called phospholipase C, and tyrosine kinase, using the enzyme tyrosine kinase. So now you've seen some of these before, undoubtedly, right? Because we talked about this in the autonomic nervous system. So if you don't remember the autonomic nervous system, it's okay. You get another chance to explore some of those activities here. Okay. So for the cyclic AMP, which uses what we call adenylase cyclase, so imagine here's the receptor for that hormone located on the cell membrane. Okay. This receptor is what we call a G protein coupled receptor, because there is a G protein that is coupled to this receptor. So this receptor has a high affinity for whatever this hormone is. Once the hormone binds to this receptor, it activates this G protein, and the G protein dissociates. Once the G protein dissociates, it will dissociate into an individual subunit. So you guys don't really have to worry what kind of subunit that is. But the subunit slides over and activates this enzyme called adenylase cyclase, which is located right next to this G protein coupled receptor. Adenylase cyclase, its job is to remove 2-phosphate from ATP. So you have adenosine triphosphate, which is ATP. You take 2-phosphate away, now you have adenosine monophosphate. But adenosine monophosphate will immediately reconfigure itself into a cyclic form. We call cyclic adenosine monophosphate, or cyclic AMP. Okay. The cyclic AMP is going to activate what we call a protein kinase. And that protein kinase is in its inactive form at that point. So once cyclic AMP binds to it, or binds to whatever it is, or remove this regulatory unit, or whatever the case is, then the protein kinase becomes active. So all we need to know at that point is that cyclic AMP is going to activate your protein kinase. Okay? And this protein kinase, because it is a kinase, what it does is that it will remove phosphates, 1 phosphate from an ATP compound, and take that phosphate and sticks it onto an inactive protein or an inactive enzyme within that cell in order to activate it. Okay? So the job of protein kinases is to phosphorylate inactive proteins and inactive enzymes, okay, in order to produce whatever desired effect you want to produce by the activities of this hormone. So this hormone, once it binds to its receptor, will initiate this whole chain of events to eventually allowing for proteins and enzymes inside the cell to become active, to achieve whatever functions it needs to achieve. Okay, so that's the cyclic AMP pathway. For calcium calmodulin, secondary messenger, it utilizes what we call a phospholipase C enzyme. So again, it is a G protein coupled receptor. In this case, it has a G protein coupled to the receptor. The receptor is highly specific and has high affinity for whatever that hormone is. The hormone then binds to this receptor, activates the G protein, a subunit dissociates, slides over, and activates this enzyme, which is located next to that G protein coupled receptor. This enzyme is called phospholipase C, which then takes a phospholipid and cut that phospholipid into what we call a DAG, which is diacylglycerol, and IP3, which is inositol triphosphate. The IP3 is internalized, well, both of them actually internalized, but the IP3 binds to and stimulate the release of calcium ions from your smooth endoplasmic reticulum. So add the word smooth here, because I forgot to. Okay. The calcium ions then binds to and activate this enzyme or protein called calmodulin. Then that whole complex, the calcium calmodulin complex, is going to bind to and activate your protein kinase. Now once again, once the protein kinase is active, it will phosphorylate inactive proteins and inactive enzymes within that cell. Okay. So it's just another series of cellular events that is set off via the binding of this hormone to a specific receptor, utilizing the phospholipase C as another enzyme that will send down all of these activities, which then allow for the activity of calmodulin calcium complex to activate your protein kinase. Okay. So the whole point of all this, whether it's the cyclic AMP pathway or the calcium calmodulin pathway, this hormone is trying to activate protein kinase. But it can't do so directly, so it has to use what we call a secondary messenger system, like a tag. Right? Tags this message, or messenger, and the messenger then tells whatever the protein kinase is supposed to do, so they do it. Same thing in the calcium calmodulin. So this hormone activates the secondary messenger called calcium calmodulin in order to activate protein kinase. That's the end game for this hormone. Right? That's what it's trying to do, activate the protein kinase. All right, now for tyrosine kinase. Tyrosine kinase is a very different type of receptor because in this particular receptor, it has three portions. It has a ligand binding portion, it has a transmembrane portion, and it has a cytoplasmic enzyme portion. And this particular portion, this enzyme here that's inside the cell, has a bunch of tyrosine residues, tyrosine amino acids that makes up, because tyrosine is an amino acid, right? So it has a bunch of tyrosine that makes up the structure of that particular protein. And this protein is a kinase. Hence we call this tyrosine kinase. Okay? So there's an enzymatic portion here, there's a kinase portion in the cytoplasm attached to what we call a transmembrane portion, which basically is integrated into the membrane. And then there's a portion that's outside of the cell called the ligand binding portion. In this particular example, they call it the signal binding site, right? Which essentially binds the hormone. All right, so now typically in this setup, you always have two of these particular receptors located right next to each other. They're separate at this point. So once the hormone binds to this receptor binding portion, the signal binding portion here, there's two of the similar type of hormones binding to each of these receptor sites. Once it does that, it causes these two structures to come together to form a dimer. Okay? It's not the same as the dimer that we talked about in nonpolar hormones and stuff like that. But it's a dimer because there's two things that come together to form one complex. Once it forms that dimer, it will likely activate the tyrosine kinase. Okay, so once the dimerizes, it basically activates this enzyme down here called tyrosine kinase. But the activity of that tyrosine kinase is kind of weak at this point, okay? So once that tyrosine kinase is weakly activated due to the dimerization effect, what it does then, it's gonna recruit, it's gonna bring in ATPs, and it's gonna start to remove the phosphate from ATPs and stick those phosphate onto each of these tyrosine residues. So basically, once the tyrosine kinase becomes weakly active due to the dimerization of those receptors, it will phosphorylate itself. Once it phosphorylates itself, it becomes fully active as a tyrosine kinase. Now, as a fully active tyrosine kinase, all of these inactive proteins and enzymes will basically move toward these tyrosine kinase. And as it moves to these tyrosine kinase, the tyrosine kinase will phosphorylate these guys in order for it to become fully active. So instead of the tyrosine kinase, or instead of a kinase that is inside the cytoplasm, this kinase is actually bound to this receptor right at the cell membrane. So therefore, in the other system where you have calcium calmodulin activating the protein kinase, or the cyclic AMP activating the protein kinase, which is inside the cytoplasm, those kinases do the same thing. They will take phosphate from an ATP and phosphorylate an inactive protein to activate it. This kinase does the same thing, but this kinase is actually bound to this receptor and has a bunch of tyrosine amino acid, and that's the reason why we call them tyrosine kinase. So therefore, once they autophosphorylate, they phosphorylate themselves to become fully active, then they can transfer that phosphate to those inactive proteins in order to activate them. And as soon as they transfer the phosphate to those inactive proteins, they'll pick up another phosphate from ATP and autophosphorylate so that they remain highly active. Make sense? Right. So imagine if I'm a tyrosine kinase, and now let's say once that receptor binds to the hormones and it dimerizes, I become weakly active, so I'm just gonna call a bunch of ATPs toward me. I'm just gonna remove one phosphate of the ATP, sticks it onto me to become fully activated. Okay, so now I'm a fully active tyrosine kinase. And then once I'm fully active, I'll say, all right, you inactive proteins and enzymes of this cell, well, come over here, line up. I'm gonna give you some candy so that you can go on your merry way. So like an inactive protein comes along, I take this phosphate, sticks it onto that protein, activate this guy, pick up another phosphate from ATP, phosphorylate myself, another protein comes by, I take that phosphate, sticks it onto them, activates them, and so on and so forth. Right? So that's the activities of tyrosine kinase. Any questions about that? Okay. Any questions about anything that we just talked about? Sure. All right. Okay, if you're not sure, then we're gonna jump into it. I'm gonna show you guys the GPS system that I use to navigate through all of this endocrine stuff. So, one of the main endocrine gland in the body is your hypothalamus. The hypothalamus is considered to be the master endocrine gland of all the major endocrine glands that you have. Okay? Because remember, in the hypothalamus, there's like multiple nuclei that contains a lot of different neurons and stuff like that, right? So these particular neurons all produce hormones. And some of these neurons extend out of the hypothalamus. Some of these neurons stays within the hypothalamus, but they all are stimulated to produce and secrete hormones and things like that. So therefore, there's 3 nuclei that you guys need to keep in mind that has neurons that extend out of the hypothalamus itself to move somewhere else within the regions of the brain. Okay. So one is called the suprachiasmatic nucleus, okay, which contains neurons located right on top of the optic chiasm. You guys remember the optic chiasm where the neural fibers carrying visual information crosses over and stuff like that, right? Okay. Another one is called the supraoptic nucleus, which basically is a collection of cell bodies of these neurons located on top of your optic nerves. Okay. And the third one is called the paraventricular nucleus, okay, which is located right next to the ventricles, which is a fluid-filled cavities inside your brain, right, filled with cerebrospinal fluid. All right, now the suprachiasmatic nucleus has a lot of complex connections and stuff like that. One of the connections that it will eventually produce is a connection with another glandular tissue here called the pineal gland. The pineal gland is located in what we call the epithalamus of your brain. So the diencephalon has the thalamus, which is a relay station for all incoming sensory information, except for smell. And you have the hypothalamus, which is located underneath the thalamus, and you have the epithalamus, which is located toward the back. Right? You guys remember that discussion we had of the central nervous system at some point. Yeah, all right, that's okay. Okay, so in the suprachiasmatic nucleus, these neurons will extend out, so it doesn't have a direct connection, but I'm just gonna draw it that way anyway, right? Just so you guys understand that it does eventually have some connection and some stimulation to the pineal gland, okay? Now, in order for it to stimulate the pineal gland or inhibit the pineal gland, then it has to receive some kind of information from something else. So that something else is known as the retina. You guys remember the retina? Located in your eyeball, right? The retina has a bunch of photoreceptors, rods and cones and stuff like that. What's the functions of those particular cones inside the retina? What kind of vision does it detect? Color vision? In what kind of light environment? Light or like bright light or really dim light? Right. Bright light, right? Like right now. If you see color, you're using your cones. You can't see colors, you're using the rods. And you don't use the rods during bright light environment because when it's bright out, all that photon of light inhibits all your rods. Right? You guys remember the bleaching effect and the regeneration and the bleaching effect and regeneration of those particular rods, right? All right, anyhow. So the retina is going to detect what we call the environment around us. You guys remember the visual fields and the retinal fields and all that good stuff. Once upon a time, we talked about that. Everybody was so happy about learning that kind of stuff. So imagine if the environmental cue represents day versus night. Okay. So let's say it's the nighttime, whatever the case is, right? So the night will stimulate. Well, obviously, both of them will stimulate the retina, day or night, doesn't really matter, right? Because the retina detects all of those visual, information. So if you look outside, it's dark right now. Let's say if you're driving in at about 4:30, and all of a sudden you see that kind of sun setting over the horizon, having a nice little coloration of the background in the sky and stuff like that. So then the retina will relay all of this information to this area within the suprachiasmatic nucleus. Okay? Of course, in addition to relaying that all the way to your occipital lobe in your primary visual cortex and all that good stuff. Okay? The suprachiasmatic nucleus will detect this and will determine whether or not the pineal gland should secrete its hormone. Okay? Because the pineal gland is another major endocrine gland of the endocrine system. So therefore, let's say it's nighttime or it's getting dark. The horizon, the sun setting down across the horizon. This image is relayed to the suprachiasmatic nucleus. The suprachiasmatic nucleus will basically regulate other areas of the brain and eventually stimulate the pineal gland. So stimulate during night. Okay. And when the pineal gland becomes stimulated by the neurons of the suprachiasmatic nucleus, it will release a hormone we call melatonin. Please don't confuse that with melanin, because melanin is a pigment that is secreted by your melanocytes, okay, on your skin, in the eyes and stuff like that, which gives you different texture or different coloration of your skin and of your hair and all that good stuff. Okay? But melatonin is a type of hormone secreted by the pineal gland, and what melatonin does is that it basically is going to be released out throughout the central nervous system. It's going to bind to different regions of the brain and decrease. So target of melatonin is, we'll call it CNS structures. Because we're not going to list all of them, right? And then the function, and the function of melatonin is to decrease CNS activities. And when you decrease all the neurons within the CNS, decrease all their activities, you are getting your brain ready to go to sleep. Okay? So essentially, the function of melatonin is to regulate what we call circadian rhythm of the body. Sort of like the sleep-wake cycle of the body. Okay. So let's say if you are a graveyard shift worker, when you have to work, and when you guys start out, as let's say in nursing, as nurses and stuff like that, once you get your nursing degree, whatever the case is, and you start out, most of the time you get the worst schedule, the worst hours, right? Because people want to work at a certain time or whatever it is. So you get most of the time, the most available stuff is gonna be anywhere between, 11 at night until 11 in the morning, something like that, or 9 to 9, or whatever the case is. I don't remember what the time frame was, so it depends on the clinic itself. Basically, that's the graveyard shift, okay? So you work that shift, your circadian rhythm will be absolutely abnormal, okay? At some point, you acclimate to it maybe after 2 months, and maybe after then, you switch to a different shift, and you have to reacclimate yourself to that shift, okay? Because it kind of sucks. All right, anyhow, so that's melatonin. Any questions on this? Okay, so that's the stimulation of the retina, and the retina will stimulate the suprachiasmatic nucleus. Suprachiasmatic nucleus will stimulate eventually the pineal gland to secrete melatonin during nighttime, okay, when it's getting dark out. So the retina will always stimulate this suprachiasmatic nucleus. However, when it is daytime, the image is still being detected by the retina, still being stimulating to the suprachiasmatic nucleus. But when it's daytime, the suprachiasmatic nucleus will basically inhibit the pineal gland. And because of that, it will decrease the amount of melatonin secretion during daytime, so that your CNS structures can become more alert and awake to allow you to stay more alert and awake throughout the whole day. Okay? So that's one hormone that you guys need to know. All right. In the supraoptic nucleus and the paraventricular nucleus, you also have these these neurons that extend out of the hypothalamus. And right below the hypothalamus is a glandular tissue we call the pituitary gland. The pituitary gland has two portions. There's an anterior portion and a posterior portion. The posterior pituitary gland is a direct descendant of neural tissue. So in development, you have this brain tissue on top, and all of a sudden it says, you know what, let's extend downward. So it starts to extend downward, pushes down on neural tissue. Okay, so there's a portion of it that extends downward. And down here, there's a bunch of epithelial tissue. So those epithelial tissue, as it develops, it starts to push upward. And as the epithelial tissue meets with this neural tissue, they actually comes together to form your pituitary gland. So the posterior pituitary gland came directly from neural tissue, whereas the anterior pituitary gland came directly from epithelial tissue. Okay, so therefore, it's two different portions of your pituitary gland. So the neurons coming from your hypothalamus that extends out of the hypothalamus in the supraoptic and paraventricular nuclei extends all the way down into your posterior pituitary. Okay, so you have neurons from here extending down into the posterior pituitary. You have neurons from here extending down into the posterior pituitary. Okay, so please understand that the posterior pituitary gland does not contain any cells that produces and secretes hormones. It is just storing, or it's just a place where the axons and axon terminals of these neurons coming from the supraoptic nucleus and the paraventricular nucleus in the hypothalamus that extends all the way down. Okay. All right. Now, another thing to think about is that when the hormones are made, they are made in the nuclei of these neurons. Does that make sense? Okay, that's where all the DNA structures are, because it has a nucleus within those particular cell body. It has a bunch of rough endoplasmic reticulum to basically translate all of those particular messenger RNA, so it makes all of those hormones there. The hormones are made in the supraoptic nucleus and the paraventricular nucleus of the hypothalamus, and they are transported down the axons and stored in the axon terminals of these neurons located in the posterior pituitary. So therefore, any hormones that are made within the supraoptic nucleus and paraventricular nucleus will be secreted from the posterior pituitary. They are not secreted from these areas inside the hypothalamus. Make sense? Okay. So for these particular hormones that we're going to talk about, they have a different source of production and site of secretion of those hormones. Typically, most glandular tissue, their source of production and the site of secretion will be the same. Okay. The only difference are these guys. All right. Now, for the supraoptic nucleus, when they make these hormones and basically transport these hormones and store them into the axon terminals, when they are stimulated, they will release these hormones, and the hormones coming from the neurons of the supraoptic nucleus is known as antidiuretic hormone. Antidiuretic hormone is also referred to as vasopressin. The technical name of this is arginine vasopressin because there's a lot of arginine amino acids associated with that vasopressin. Okay, so. All right, so antidiuretic hormone is secreted by these neurons coming from the supraoptic nucleus, and these hormones will be secreted from your posterior pituitary gland. Okay. What does antidiuretic hormone go to? What's the target?
Collecting duct.
Collecting duct of?
Yeah, the nerve.
Yeah. Collecting duct of the nephron or of the kidneys. That's fine. Okay, so collecting ducts. We'll call it the nephron. Technically, it's not really part of the nephron, but you know, that's all right. Then what does it do at the collecting duct of the nephron?
Reabsorb water
How does it reabsorb water?
from the aquifers.
So it increases expression of aquaporins to increase water reabsorption. Okay. That's a function of antidiuretic hormones. All right, so what do you guys think stimulate this antidiuretic hormone to be released from the posterior pituitary? What stimulates these neurons in the supraoptic nucleus? So one of the things is an increase in osmolality of the blood. What does it mean when you have an increase in osmolality?
You have low concentration of ions.
Low concentration of what? Volume?
Of ions.
Of ions? Low or high?
Oh.
So osmolality refers to the collective amount of moles or molos of those particular solids. So you have too much of those solids compared to the amount of volume, the amount of water, right? Then you have high osmolality. What does that mean? It means maybe you have too much of those things, or you just have too little amount of water overall. Okay, so if you have too little amount of water, that means that an increase in osmolality is the same thing as saying dehydration. Okay. All right, another stimulus is a hormone. Do you guys remember what hormone that would be?
Aldosterone.
Not aldosterone. It doesn't go to the brain. It's part of that pathway. It's part of that system. It's part of that RAS. What's the middle one? Angiotensin II.
Oh.
So angiotensin II also stimulate these particular cells, these neurons within the supraoptic nucleus. So because it's a hormone, I'm going to circle it in red. Stimulates those neurons directly so that they can release antidiuretic hormone from the posterior pituitary gland. So that takes care of that hormone.
So wouldn't be the juxtaglomerular apparatus
Well, that thing is just so far-fetched. Where is it located?
in the kidneys?
Okay. And then what does the juxtaglomerular apparatus stimulate or release?
Renin.
Okay, so that's what it does. And what does the renin do?
Turns angiotensin into angiotensin
Okay. And then what converts angiotensin 1 to angiotensin 2?
angiotensin converting
Okay.
enzyme.
So therefore, that's like 4 steps away, right? So you can't say the kidneys directly stimulate the supraoptic nucleus.
But would the increase in blood osmolality directly trigger the supraoptic
It does.
nucleus, or because I remember from what we learned that increase in osmolality is
It's one of the
detected
ways.
by the
No. That increase in osmolality is not detected by the kidneys,
oh
right? That increase in osmolality basically means you have a low blood volume. If you have a low blood volume, what happens to your filtration rate?
increases.
Decreases.
Oh,
Okay, so you have a decrease in filtration rate, what happens to the amount of filtrate? This is how you tie all of this into the kidneys as well, right? So you have a low filtration rate, you're gonna have a low volume of filtrate. At the proximal convoluted tubules, 85% of that is taken back already. Immediately, once you get into the nephron loop, you're gonna have the countercurrent multiplier system kicking in even more. Because now you know your body is dehydrated, so the kidney is taking as much back as possible. Whatever is left now moves into the distal convoluted tubule. So because the proximal convoluted tubules and the nephron loop has already taken back so much of that stuff, by the time the overall filtrate volume reaches the macula densa, which is part of your juxtaglomerular apparatus in the distal convoluted tubules, whatever that volume is that reaches that area is now lower than what it should be. It should be low under normal situation, now it's actually even lower. So the macula densa sees that and says, I don't like that. Why do we have such a low filtrate volume? Why is it that other parts of the kidneys are taking back so much more water and solids? So because we have low filtrate volume, that must mean we have low blood volume. So we have low blood volume, I have to do something to increase that blood volume. So then it stimulates the granular cells to secrete renin. And then renin will activate the overall renin-angiotensin-aldosterone system. Right? And aldosterone or angiotensin has this particular effect at the supraoptic nucleus to stimulate the release of antidiuretic hormone from the posterior pituitary so that it can stimulate the collecting ducts of the kidneys to increase the amount of water reabsorption back into the body. So all of that basically is sort of like an effect of all of those hormonal regulation. Because you want to take back as much of that contents from the kidneys as possible. Otherwise, you're gonna lose it as urine. Make sense? Alright. Good. Okay, that's why we talk about this in the cardiovascular system. So we talk about this in the urinary system. And now we're gonna talk about this, well, we just kind of talked about it in the endocrine system. So you guys should know it by heart at this point. Okay. And what's the effect, the overall result? Once you have that renin-angiotensin-aldosterone system kicking in, what's the overall result in the body? What's it gonna do? What is it gonna do to blood volume? Increases. What's it gonna do to blood pressure? Slightly increase that as well. Okay? And what's the one that stimulates blood pressure increase? Angiotensin 2. Because it vasoconstricts some of your peripheral arterioles. Okay? Vasopressin is called vasopressin because it has those slight constrictive effect to the smooth muscle as well. But it's not as effective as the other hormones. So it does have some degree of that constriction, therefore slightly raising the pressure within your systemic circulation to slightly raise your blood pressure. Why do you need to increase your blood pressure? What's Ohm's law? Increase the blood pressure, increase the flow of blood. You want that flow to get back into different organ systems of the body, and one of those organs will be your kidneys, so that you can satisfy those kidneys. And say, all right, you can shut down the renin-angiotensin-aldosterone system now because we don't need to take back any more fluid from the kidneys. So it is connected. All right. Now the other area, the other neurons coming from the paraventricular nucleus is going to release a hormone from the posterior pituitary, and that hormone is known as oxytocin. Okay. Oxytocin goes to two main places. There's two targets for oxytocin. So, targets. Number one, it's gonna go into the uterus of the female reproductive tract. And that portion of the uterus contains a lot of smooth muscle. So, we'll call it the smooth muscle of the uterus. And I'll give you the terms of this. This is called the myometrium. Okay, because we'll talk about that later on in the reproductive system anyways. Okay, so the myometrium is where the smooth muscle of the uterus is located. Okay. Now, I'll put the function down here as well, just to match it up. So the function, the first one here, as oxytocin stimulates the smooth muscle of the uterus, what do you think it's gonna do? So increase the contraction of the smooth muscle. So increase smooth muscle contraction for what we call labor and delivery. Okay. So therefore, what is the first stimulus at the paraventricular nucleus? It will be labor. Okay. When it comes time at the end of the pregnancy, in order to evacuate that little kid out of your body, because it's, kid's been growing already, it's time for that kid to suffer like everybody else and stop relying on his mom. Well, maybe not like totally unrelying on his mom, but no longer living inside the woman's body and stuff like that, just feeding off her nutrients and oxygen, right? So it's time to deliver that baby out. So during that time, because the head is now pressing onto the cervix, which is a part of the female reproductive system, causing or stimulating inflammation, irritation and inflammation, and therefore the cervix starts to dilate out. And all of these particular processes stimulate smooth muscle contraction. That's what we call labor. And that smooth muscle contraction is detected and relayed all the way back to the paraventricular nucleus of the hypothalamus, which then stimulates it to release oxytocin from the posterior pituitary. Oxytocin then goes all the way back in the blood, it goes all the way back into the smooth muscle of the uterus, stimulate that smooth muscle to contract even harder, right? That increase in contraction is going to eventually expel that kid out from the uterus itself. So we're trying to deliver that child. All right, the second target of oxytocin is basically what we call the smooth muscle that lines the duct of the mammary glands. So smooth muscle of what we call mammary ducts. Okay? Because the mammary gland is a gland that produces milk, and it's an exocrine gland because you need to secrete that milk into the environment around you. So therefore, that exocrine gland is attached to a bunch of ducts. And these ducts has a bunch of smooth muscle that surrounds it. So therefore, oxytocin is gonna stimulate the smooth muscle of the mammary ducts to increase smooth muscle contraction for the purpose of milk ejection. Okay, so now you wanna get that milk out and eject it out. So smooth muscle contraction is gonna help to mobilize that milk from the mammary glands out into the environment around it. So therefore, this is for the purpose of feeding your newborn, right? So one of the way to stimulate that increase in ejection of the milk, that smooth muscle contraction, is for the baby to latch on to the mom's nipple. So basically, once you have that newborn, right, the kid is delivered, they cut the cord, they, clean them up, put them in under the heat lamp, figure out the Apgar score just to make sure the baby is fine, active, breathing fine. adequately perfusing all over the body and stuff like that. And they wrap the baby up in a blanket and they give him back to the mom. Okay, the first thing that they want to have the mom do is basically maintain eye contact with that baby to establish that emotional connection. Okay, and of course now the baby's out, most likely the baby's hungry, so you gotta teach the baby how to feed themselves, right? One of the things that you do is that you take the baby's mouth and you put it close to the mom's nipple. And you try and stimulate the baby by brushing his cheek or whatever, we call it the rooting reflex, and allowing for the baby to seek out that nipple and latch onto that thing. Once the baby latches onto the nipples, the baby will start to suckle. This is basically a physical activity stimulated by, I don't know, its buccal surfaces and all the muscles there. So that suckling is going to stimulate the release of oxytocin. And that oxytocin will stimulate the smooth muscle of the mammary ducts to contract. And once that smooth muscle contracts, you will now have an increase in milk ejection. So therefore, that's one of the hardest things oftentimes for new moms. Because the baby may be fussy or the baby may not be able to latch appropriately onto the nipple. And if they can't latch onto the nipple and they try to suckle and they disengage from that nipple, they'll just suckle like the air and stuff like that, right? And nothing's coming out, and the baby's crying even more because like, hey man, where's my food? Kind of thing, right? And the mom gets frustrated and stuff. So you gotta teach the mom, or tell the mom to be patient and kind of show the mom appropriately how to adequately, like, don't press the kid's head onto your breast and stuff like that. You don't want to suffocate that kid, but you want to stimulate that kid to a point where they can actually seek out and easily latch onto the nipple and basically start that suckling process. Okay? So therefore, if you guys ever work in the OBGYN clinic and the newborn clinic and stuff like that, you're gonna have to be patient and teach the mom that. Okay, and then of course, other things that you need to watch out for is what we call postpartum depression. Because for some reason, when the mom delivered that baby, the mom feels a disconnect to the baby because the baby is no longer part of her. Like, imagine you carry this living entity for like 9 months, right? Everything you do, the kid is moving around, kicking every single internal organs that you have. Like, hey, that's wonderful. Not really, but it's wonderful in some sense, right? So therefore, once you give birth to this child, it's a traumatic experience too. And this child's out. Now the child is like, oh man, a separate entity from you. So for some reason, a lot of the newborn, the new mom has this weird kind of depressive feeling that there's no longer a physical connection between the mom and the child anymore. And for some reason, the mom stops caring for that baby. And oftentimes, in severe cases, can lead to killing that baby himself. Okay, so you gotta watch for some of that too. All right. Now, of course, you can't do that, obviously, but you can always check up on the parents and stuff. Okay, good. Now, oxytocin is not only a hormone that benefits female individuals, because you can see these two particular targets that I've listed out here are only in the female individuals that can have the ability to, carry the child in pregnancy, right? Oxytocin is actually considered to be your primary intimacy hormone. Intimacy does not mean sexual intercourse or sexual activities of any kind. Intimacy is defined as the sense of belonging, okay? So let's say if you guys are watching the Niners game and we're all like Niners fans and stuff like that, and the Niners start to beat the crap out of the Panthers, and we're all like, yeah, cheering and high-fiving and, chest bumping and hugging each other. When you do that, oxytocin is released. Because now you have a sense of belonging to each other. We have a common ground, a common interest, right? Even when you're walking down the street and you haven't seen your buddy for a long time, you come across this guy, it's like, wait a minute, I know you from high school, right? Like, that was a long time ago. Hey man, how you doing? You give that person a hug, oxytocin is released. Okay? So anytime you have that basically sense of belonging to another person, to another group, being recognized, being appreciated, and stuff like that, oxytocin is released. Okay? And oftentimes, if you are not around people or not, like a lot of people would like to be secluded, a lot of people don't like to have like partners or people around them, they don't have a lot of oxytocin, and they are more prone to kind of like what we call non-community type of behavior, right? A little bit of that apprehension about being around people and anti-community and stuff like that, right? So therefore, it's helpful. It's healthy to be around people all the time, to interact with people oftentimes, okay? So if you guys have, let's say if you guys are married or you guys have a partner and you know she's pregnant or whatever, or she's just delivered the baby, or whatever the case is, and you know you go off to work, you go off to school, and you come home and you're like, all right, I'm tired, I don't want to talk, I don't want to do anything. Take a moment to understand that that person also require some degree of interaction as well, right? So therefore, that's some of the things that you can also educate your patients on if they are new parents. Let's say one of the partners stays home and take care of the newborn, the other partner goes to work. So therefore, once the other partner comes home from work, make sure you advise them to take some amount of time for each other, because that will help to maintain that healthy mental state, okay? That intimacy type of feeling inside of all these individuals, right? Otherwise, then they're more prone to depression, and they can become more withdrawn and stuff like that from their partner. Okay, so very important that's going on. Okay, now of course, that being said, oxytocin doesn't mean that it's not a vital part of sexual interaction, because oxytocin oftentimes can also enhance what we call sperm mobility and also oocyte mobility. And it's also very important in the female individual to achieve what we call female orgasm, because female orgasm is about contraction of the uterus. And let's say during sexual intercourse, if you're trying to conceive a child, and the male individual ejaculate into the female receptacle, then if you have the uterine contraction, this will help to mobilize the sperm much faster toward that egg, so you can fertilize that egg with more efficiency. Right? So anyway, that's oxytocin. Any questions? Okay, that's only 3 hormones. Let's go to that. Well, now I'll give you the other 40.